Welcome everybody to another Authors at Google talk. Today we have as our guest at Google Professor David Deamer from the Department of Biomolecular Engineering at the University of California. He's going to talk about the evolution of very early life, and his book, which just came out in print, is called First Life. So please welcome David Deamer. Thank you.
Well, thank you all for coming. It was really a treat and a pleasure. I gave a tech talk here last year, but what you're going to hear today is really my first love, which is how did life begin and how did life begin to evolve? So we're making a lot of progress, and you're going to hear about that today. You're also going to hear a little bit of history about how my own research career got started and how it's progressed through the last about 30 years, believe it or not, and how it finally culminated in this little book that came out earlier this month. So this is an author's talk, and we're supposed to do little readings out of our book, but that's kind of boring just to hear readings. So I'm only going to do a couple of short paragraphs here and there to illustrate as we go on. So First Life, that is the book company's UC Press title. I thought it was pretty good. We played around with a lot of titles, and that was the one that really came out. I later discovered that this was also on David Attenborough's television series, so that's just luck of the draw. So First Life, and what you're going to hear about from me today is that life, at least at its beginning, and still today, depends on what is called self-assembly processes. And what you're seeing up there in that little image to the right is a bit of dried phospholipid. And when phospholipid interacts with water, it self-assembles into membranous structures. So as part of my talk, I'm going to take you back 40 years in time to the person who discovered this and how I spent some time with him and began to work on this project. So it's not just this book that came out, which is sort of a lay person's version. It's readable, I hope. There's also a technical version that came out last year. Jack Sostak and I got together with the Cold Spring Harbor Press. And this is a 20 chapters from a variety of authors, including Jack and myself, giving a very up-to-date vision of how life could have begun, right from the start, in fact, as Earth became a planet. So those are the things that you're going to hear a bit about today. And I'm going to start by reminding you of this saying. Most of you know the acronym. It's keep it simple, stupid. Leaving out the stupid, this is what we tell our students in our laboratories. Keep it simple. And what that means is it comes out of a reductionistic philosophy that scientists use to try to literally keep it simple, to keep things under control. But I have another little acronym that I pass along after I get that across, BNTS, and that stands for not that simple. Because we have been limited, we are limiting ourselves as we think about the origin of life by doing little baby steps, this molecule, that molecule. How do they link together to make a little dimer of some sort? So these are baby steps. I think the point of my book is let's look at a bigger picture and try to get something sufficiently complex. Well, sufficiently complex, what do I mean by that? Some of you may recognize Stanley Miller's experiment that was published about 1953, however many years ago that is, half a century to round off. And what Stanley did was to put together a sufficiently complex simulation of the early Earth. And this is how we learn about the origin of life. We try to simulate something in the laboratory, keeping it simple, and try to learn from a simulation that tests a hypothesis. So Stanley's hypothesis was that if you had four gases, water, ammonia, methane, and ammonia, those four gases, the four biogenic elements, and if you put in some energy as a spark, an electrical discharge there, that would be sufficiently complex to make something interesting. Those are the four molecules, very simple gas molecules, including boiling water down there in the bottom left. And he then sparked it for a day and for a week And he had this spark discharge going, and it began to turn sort of a light yellow color, and then an orange color, and then a red color, and then a brownish color about a week later. And the sides of the flask became coated with the product of this reaction. Amazingly, there were hundreds of products produced from this simple reaction, including amino acids. And that's what caught people's eyes, because amino acids are the monomers of all life on the earth. They're used to make proteins. Smaller proteins are called peptides, and the bond that holds them together is called the peptide bond, which is one of the things we have to figure out how that came to be. So that really caught people's attention, and it started the field up. Now let's see what I mean by insufficiently complex. Suppose that Stanley thoughtlessly left out one of those molecules. Nothing would have happened. The simulation would have been insufficiently complex. And so in our laboratories today, we're trying to learn how to make our simulations sufficiently complex, and that's what you're going to hear about today. And you're going to hear about some results from this step toward the end of my talk today. Well, the concepts that I put together in First Life to produce something that I feel is sufficiently complex are as follows. The conditions, I feel that there must have been compartments. And that really is a fairly new concept. People did not think about life as beginning as cells. People thought of life as beginning as molecules. But my claim is that we really needed a cellular compartment to keep everything together in one place. There must have been a way to concentrate the organic molecules that are going to react, and they must be cycled. That's another thing we don't think about when we do these simulations. We just turn on the spark and wait to see what happens. Or we add a couple of reactants and wait and see what happens. That's the chemist's way of doing things. But there is another process, which I'm sure some of you have heard about, called the polymerase chain reaction, PCR. It depends on cycles. You put in a little bit of DNA, you cycle it through a hot-cold cycle, a little bit more DNA is produced, another hot-cold cycle, 40 or 50 cycles, and you have millions of times more DNA than you started with. This is a central process that is used in all molecular biology laboratories today, discovered about maybe 30 years ago now. So keep in mind cycles. I think the cycles are important. There must have been a fluctuating environment. The components we're talking about, of course, there must have been a source of organic compounds. The first part of my talk, I'm going to tell you about one possible source, which is an extraterrestrial source. We think that a lot of the organics that were brought to the Earth, available for the origin of life, were delivered here in the late accretion of our planet, and you're going to hear about that. Then there had to be a source of energy. You can't get reactions to go unless you go from uphill to downhill. There has to be a source of energy to get the reactants uphill. Only then can they react and go downhill toward equilibrium and make something interesting. So we have to think about what energy source was available on the early Earth to drive these reactions. Finally, sufficient complexity. That's going to involve self-assembly of these molecules, and I'm going to demonstrate this to you. It involves an organizing matrix. Now, this is a relatively new idea, by the way. We have thought that probably clay might have been involved. The surface of clay can organize molecules and allow certain polymerization reactions to occur. But I'm going to tell you about a different organizing matrix that we are working on. As a result of all this, you get what I call combinatorial chemistry. Each of these compartments is a microscopic test tube, and each compartment is different from all the others. It's a combination of molecules in those individual compartments. And I'm talking about vast numbers of compartments that would have been around on the early Earth. A few of these, a very few of these, likely had a set of molecules that by chance could grow by polymerization. They could capture energy and nutrients from the environment and undergo polymerization. Furthermore, not only grow, but they can reproduce by some process. And right at the end of my talk, you're going to hear about a way to produce long nucleic acid strands and get them to reproduce under simulated prebiotic condition. So that's where I'm going in this talk. Okay, now I want to talk a bit about how I got started in all of this, and that comes to my first reading. There's only 2 of these, so don't worry. During the spring and summer of 1975, I was spending a sabbatical leave with Alec Bangham in his Babraham laboratory just south of Cambridge, England, when an idea emerged that guided me into research on the origin of life. A few years earlier, Alec had discovered the self-assembly properties of phospholipid and showed that lecithin extracted from egg yolks could produce microscopic vesicles, ultimately forming structures that we now call liposomes. One summer day, Alec and I were driving to London in his beloved Morris Mini. We had pulled over to the side of the road to enjoy some cucumber sandwiches, I'll never forget that, prepared by Rosalind Bangham, that's Alec's wife. The conversation turned to a lecture that Alec had given a few years earlier at Bristol University with the title, Membranes Came First. Alec argued that the self-assembly properties of lipids were such that the membranous vesicles must have been present on the early Earth long before nucleic acids and proteins came along. But then we were stumped. What was the prebiotic equivalent of lipids that can self-assemble into membranes, and where did they come from? Well, here is Alec's image. This is from 1971. He let me take a picture of this. And back in the old days, before PowerPoint and Keynote and video projectors, if you were giving a scientific talk, I'm sure you won't believe this, you had to make a drawing of what you wanted to talk about, take a picture and make a slide of that. That's the way it used to be done. These days, of course, we've left that far behind. I can still remember the thrill I had when I gave my very first computer-aided talk like this. Makes things so much easier. In any event, notice that Alec has the Earth. He's got a very thick ocean. He shows evolution going around in a cycle. He shows sodium and potassium ions in the ocean. And he shows a monolayer. Look right down there at the bottom. And you can see a lot of little squiggly tails. Those are hydrocarbons. The head group is hydrophilic, water-loving, so it sticks down into the water, dissolves in the water, but the tail cannot dissolve. Oil and water don't mix. So this is a monolayer. Then he said, if you have some sort of aerodynamic perturbation, just wind, tides, waves action, and so forth, that those monolayers would crimp inward and turn into liposomes. So that is his idea about why membranes could come first. And really, from that moment on, I've been testing that idea. I'd like to show you Alec Bangham. This is a picture I took about 7 or 8 years ago. And there you see him making self-assembled amphiphilic membranes in his backyard. Those, by the way, are called soap bubbles in common language. And really, a soap bubble is a membrane. It's a self-assembled membrane. I would guess everybody in this room at one time in your life has blown some bubbles like Alec is doing up there. What I'm going to do now, though, is to show you what Alec saw under the microscope when he first discovered the self-assembly property of phospholipid. He did not have the jazz piano background, by the way. So that's what it looks like. When I do this work in my lab, I can watch lipid reacting with water to produce these beautiful self-assembled structures. Our claim is that something like that occurred on the early Earth. This is what Alec saw in the electron microscope. This is just an accident, by the way. He wanted to test out their new microscope, and he had these interesting self-assembled structures, so he put that under the microscope and added what's called the negative stain that lets him see literally at the single molecule level. And he saw not just globs, but he saw those layers. You can probably count 10 or 20 layers of in that structure. This is a multilamellar phospholipid structure, and he called these in his paper multilamellar smectic phospholipid mesophases, which is a bit of a mouthful, so people began calling them bangosomes. in his honor. And Jerry Weissman, a very early visitor to his lab before I got there, invented this word, liposomes. You may have heard of liposomes because their properties were not lost on the cosmetic industry. Christian Dior captured liposomes without telling Alec, by the way. Alec learned about this because his wife, Rosalind, received an ad like this in the mail saying that they had this cosmetic called Capture XR60/80 by Skin Extra Vital Restoring Serum. And look there, powerful liposome system. So this is now used to deliver cosmetic materials to skin surfaces, anti-wrinkles and all that stuff. It's also used in the drug industry. There are anti-tumor agents that are captured in liposomes, injected into the bloodstream, carried to the tumor cell, and produce produce death of the tumor cell. So liposomes are very powerful and far beyond what I'm telling you about today with the origin of life. But I have always been amused by that little story. Well, that brings me to the next little reading, my last one. In the summer of 1981, a small black stone wrapped in aluminum foil changed the course of my life. About the size of a marble and indistinguishable from any other rock that might be found on a beach, this stone had traveled from southeastern Australia to the NASA Ames Research Center in Mountain View, California, literally just down the street from here, where researcher Sherwood Chang showed me the specimen and gave me a small sample. But the stone had traveled even further. It was a piece of a meteor that lit up the night sky over the town of Murchison, Australia in September 1969. The fall began with a bright orange fireball. Rolling thunder followed minutes later by a shower of black stones strewn over 5 square miles. I held in my hand a genuine rock from outer space. But there was more to this meteorite than just the mineral content typical of other stony meteorites. The first stones to be discovered just hours after they fell were still emitting a smoky aroma from their hot surfaces. And the first time I ground up a marble-sized sample of the meteorite, a strange penetrating odor rose from the mortar, simultaneously smoky, dusty, and sour, reminiscent of a cigar butt or the contents of a vacuum cleaner bag. It was a distinctive smell that I would now recognize anywhere. It was the odor of outer space, nearly 5 billion years old, the aroma of organic compounds that were delivered directly to the Earth during the birth of our solar system. Nothing could be more convincing of the link between organic processes and life on the Earth. So, this is NASA Ames as it looks today. There's a nice name given over to the building that Sherwood worked in and that I've worked in, the Astrobiology and Life Sciences Laboratory. My first visit, 1981, and this is what Sherwood looked like back then as a young man. He's now retired. He's got his own ranch up in far northern California. And this is the kind of thing that he gave to me. Not this particular piece, this is a piece I got later, but he gave me a little chunk, about that big, to work with. So I'm a lipid biophysicist, and I was still trying to think of a way to test that question that Alec and I thought about. What were the first kinds of lipids? So if you're a lipid person, you use a organic solvent to extract what you're interested in, and nobody had tried this before. So I treated a chunk of that murchison with chloroform and methanol, and you're going to hear about that as soon as I tell you just a bit about where it came from. So one source of organic compounds on the early Earth is delivery during late accretion. So this is the asteroid Eros, and it's from a NASA JPL flyby in the last few years. Look at the impact craters. Those are on every asteroid we've looked at. Those impact craters occur when a smaller chunk of stone hits the larger chunk, and we've actually seen that happen. Just by chance, this image was captured when people were looking at the asteroid belt, and they saw what looked at first like a comet, but it wasn't. It turns out that tail coming out of that little white speck there, you can see in the enlarged inset, was in fact potential meteorites. Any of those chunks that happened to hit the Earth would have been a meteorite. And this is how the Murchison meteorite got here. So we are seeing with carbonaceous meteorites samples of extraterrestrial material that probably came about the same time as the birth of the Earth. And it's been delivering organic compounds to the Earth ever since, as you saw in that Murchison meteorite. This is what it would look like if you happen to look up in the sky. This is just a Photoshop version that I put together for illustrating purposes. But there was an explosion up there, and the pieces fell to Earth in the little town of Murchison, Australia. This is a real image of the town of Murchison that you can see in the upper left corner. And each of those red spots represents a place where a piece of the Murchison, maybe about this size to this size, was picked up, along with hundreds of smaller pieces. Those are the ones that I've worked with. So those are in museums all over the world. They've been worked on extensively by the community with an interest in this, and we know more about the Murchison than any other meteorite so far. So this is why they're so interesting. If you analyze the Murchison, you discover that it's around 2% organic material by weight, most of which is a kind of a polymer called kerogen or kerogen-like polymer, but some of them are soluble, that little red pie slip there. And then of the soluble, you can see that some of them are carboxylic acids. There are dicarboxylic acids, amino acids, amides, aliphatic hydrocarbons, aromatic hydrocarbons, aldehydes, alcohols, amines, and even purines. You know that amino acids are what we use as monomers for proteins. Purines, like adenine, are used in nucleic acids. So here is literally all or most of the stuff that we would want to have available if we're going to think about how life could begin. The one I'll want to call attention to is the carboxylic acids, which are about half of the mass of the organics. And one of them that you're going to see is a membrane-forming compound, because that's what we discovered. And here it is. So I extracted a chunk of the Murchison, maybe a gram sample, put it into a mixture of chloroform and methanol, ground it up, took the sample and put it under a microscope. You let the chloroform dry. And then you add water, which is the way you watch self-assembly, and this is what I saw. Really quite extraordinary. This stuff has never been alive, never part of the life process, but it still could self-assemble into membranous vesicles. And we have proposed that these are the kinds of compartments that would have been available on the early Earth for the first living cells to take advantage of as a house to live in, as they did the rest of what life is all about. So here's a phase image of some of the vesicles. We captured some fluorescent dye in it. That's what you see over on the right-hand side there, just to demonstrate that these are actually compartments that can contain something. And what you're going to hear a little bit later is that these compartments have some very special properties helping polymerization to occur. Well, what are those membrane components? One last trip to Stanford, and we took some of that sample to Dick Zayer's lab. He's got a beautiful mass spectrometer to work with, and he sent me back this image, which shows that what that stuff is octanoic acid, nonanoic acid, decanoic acid, and all the way up to 13 carbons long. This is what those things look like, by the way. In a membrane, you have the hydrocarbon tail, the black and white, and you have the hydrophilic head group. This is nothing but soap. You wash your hands with this, cleans it off. This is a soap molecule. So we had found soap molecules in the Murchison meteorite and discovered that they could self-assemble into those membrane structures that I showed you there. Really quite extraordinary. So there's what those things look like. They've been stained with the fluorescent dye. That's just decanoic acid. That's this stuff, just to prove that it can form membranes. Well, before I go to the next step, I'm going to do a little demonstration for you just to nail down in your minds how simple the formation of membranes encapsulating stuff is. So I used to be a teenage magician, and this is as close as I get these days to doing a little bit of magic. Here's just a buffer solution, water, plus a little bit of buffer in it. Here's a nucleic acid. This particular one is ribonucleic acid, but I could use deoxyribonucleic acid as well, DNA. So I'm going to put a few milligrams in here. Dissolves right away. It's water soluble. And the question is, how could something like this, even if it got synthesized by some process on the earth, how could it get into these vesicles I showed you? Because vesicles are relatively impermeable to big molecules. There must be a mechanism. And if you just do nothing more than dry down your lipid, mix in your solution, and shake it up, what you discover is that you have self-assembled the lipid vesicles. That's that cloudiness you see there. And the lipid vesicles have encapsulated the RNA. It's as simple as that to make what we call protocells. These are steps toward the origin of life, biggish molecules with potential catalytic replicating ability trapped inside lipid vesicles. So that's how simple it is. So we're not saying anything outlandish when I claim that it's easy to do and easy to conceive of. Now we're actually trying to see whether that does any good with respect to the origin of a replicating catalytic system. Well, I wanted to take you one more step in the history of this. I visited NASA Ames again in the early 1990s, and by this time we had shown that the meteorite membranes could be produced from the Murchison components, and Lou Amendola was an astrochemist. He's still active at NASA Ames. And Jason Dworkin had just finished his PhD with Stanley Miller and wanted to do his postdoctoral work in the astrochemistry laboratory with Lou. So we decided to see whether the stuff that Lou was synthesizing as a simulation of what goes on in what is called pre-cometary ice. And you're going to understand that. I got to show you a few more things before you understand it completely. But what he does is to cool a chamber, a vacuum chamber, down to liquid helium temperatures. And then he injects gases into the chamber, including water, methanol, carbon monoxide, and ammonia, and a thin layer of ice forms on that ultra-cold surface. Then he bombards that surface with ultraviolet radiation. And this is what happens in what we call molecular clouds. Our Earth, our solar system, and stars are all born in dense molecular clouds, light years in dimensions. And you're going to see a little bit more about that momentarily. And as this occurs, that ice with the UV impinging on it can make big molecules. And here's what we then published after a couple of years of work. Self-assembling amphiphilic molecules, synthesis of simulated interstellar pre-cometary ices. So the answer to where this stuff in the Murchison came from is that it was, a lot of it anyway, was synthesized on the surface of these little dust particles that I'm going to show you in just a minute. So this is the kind of image that we saw, starting with nothing more than water, methanol, carbon monoxide, ammonia. And when you hit this with UV radiation, it produces complex molecules. And I would not be able to tell you, if I had seen that image on the right, that fluorescent image, I would not have been able to tell you, is this the Murchison material or is this the synthesized material? They're really very similar in their properties. So that really gave us yet another answer. So I'm going to summarize now the first half of my talk. This is what a dense molecular cloud looks like. So we call it interstellar dense molecular clouds because they're between stars and they're dense rather than being diffuse. They're mostly made up of dust particles. These are little silicate mineral dust particles, micron size range, and hydrogen. That's what composes those. Star formation, that's where stars are produced, and solar system. The stars early in their career begin to make hard UV radiation, and that then is what illuminates the dust particles and drives the kind of chemistry I've just shown you. So let's show you a little summary of this. There's that dust particle. It's about the size of a bacterium, by the way, just a few microns across. So nothing but a silicate mineral, olivine, pyroxene. These are ordinary minerals, but it's spewed out by stars, supernova and nova when they explode. The dust particles accumulate ice mantles. The ice mantle begins with very simple compounds, water, methanol, carbon monoxide, and ammonia. UV radiation is the energy source that drives the synthesis of all that stuff on the right-hand side, including amino acids, ketones, amides, quinones, and amphiphiles. An amphiphile is like a soap molecule. Those then accrete in the early solar system to form comets, planetesimals, bring in the organic stuff along. Those then accrete to form planets like our Earth and deliver material to the early Earth. But, one little problem. The early Earth went through a collision with a Mars-sized object. This melted the early Earth. It volatilized all of the organic material that might have been there, and so we are left again with the question, where did this stuff come from? There must have been lots of stuff delivered during this accretion of the early Earth, but here now we've melted it to volcanic temperatures. That completely destroys all organic material, so we have to think a little bit further. And here's what we now think. So there's a molten moon forming from this debris ring. There's the molten Earth. The organic material has been vaporized. Sooner or later, the Earth cools, oceans form, but there's none of that original organic material. So what we think is that comets and meteorites and what we call interplanetary dust particles that have organic material on them all helped produce this last accretionary process that gave us a lot of the organics we need. So you can see the record of this in the moon. Look at it through a telescope. You see all the craters remaining from about 3.8 billion years ago. And we know that if the moon got hit, the Earth got hit, and all of that stuff is delivering organic material and water back to the early Earth when this was happening. By the way, the ocean is probably in the range of 10% comet water. A lot of the water in the ocean came out of the Earth's interior during the volcanic outgassing at that time. So that's sort of, as I say, the first half of what I want to tell you about. Just to drill this in, this is a comet that we've seen recently. We've had another probe go around the comet in orbit. It's taking these beautiful pictures. This is called the Tempel 1 comet. It's still not the comet that we can see. It's pretty far out in the solar system. When we analyze comets, we see that they're about 60% water ice, 30% mineral particles, and 10% organic compounds. So just imagine now, these are delivering water to the oceans on the early Earth. And some of this organic stuff survives the impact of delivery and would have been available for this process. You don't get a sense of how big comets are, but what I did here is to put the skyline of Manhattan to give you several kilometers in diameter. This is what comets really look like when they go past, and we can see them again. Okay, now let's go back to 4 billion years ago and think about what the early Earth was like. Here's an artist version of the early Earth. And if you didn't know that was the early Earth, you might think that that was one of the islands of Hawaii, like the Big Island. Because the early Earth was volcanic. A lot of the land masses poking up through the salty ocean were volcanic in nature. And so we have a nitrogen and carbon dioxide atmosphere. The nitrogen was still around at that time, very unreactive. The carbon dioxide was many-fold greater than it is today. Most of that carbon dioxide is now turned into limestone. But back then, that was our atmosphere. No oxygen to speak of. There's a lot of energy available, thermal energy, light energy, salty ocean. Because there were land masses, there would be freshwater ponds by precipitation. There would be electrical discharge, yet another form of energy. And that's what we had to work with. As you know, in our minds, we say to ourselves, that's the early Earth. Well, what can we do with that concept? Let's think about what was needed back then to get life started. Conceptually, pretty simple. You needed some monomers that could form polymers. We know that amino acids are in meteorites, so we imagine that amino acids were being sprinkled down or synthesized on the early Earth. We know that nucleobases like adenine are in meteorites, so we imagine there might have been ways for nucleobases to be delivered or synthesized. We know carbohydrates are in meteorites. That's work of George Cooper, just right up at NASA Ames. And we know there must have been phosphate there. So we're sort of on the way. This is what we imagine would necessarily be there. then I say that there must have been amphiphiles to produce these compartments that I've told you are so important. When I talk about self-assembly, I want to make sure that you understand what I mean by that. Most of you probably know that DNA is a double helix. You can melt the double helix, which is, a double helix. You can melt it into single chains just by raising the temperature. And it doesn't have to be very high, maybe 70, 80, 90 degrees, and you've melted the double helix. If you then cool it down, those two single strands find each other. They wind back up into the original double helix. That is a self-assembly process. Nothing makes it happen. It just happens because of the properties, the chemical, physical properties of the nucleic acids. When a protein is synthesized, it's coming off the ribosome. There's no folding, but as it comes off, it folds into a specific structure. This is essential for life to occur. If that folding were not very precise, we could not be here. That's another self-assembly. Finally, the one I've shown you is the lipid self-assembly into membranes. Well, I wanted to challenge myself and my colleagues to think beyond the laboratory. Question is, I've shown you self-assembly occurring under laboratory conditions, and I claim that it might happen on the early Earth. Is there any way I can go beyond just a hand-waving claim? Well, I think what we have to do is to try our experiments in a natural prebiotic analog environment, such as Bumpus Hell. Bumpus Hell is a few hours drive north of here. It's a little volcanic outbreak on Mount Lassen, and it's amazing to visit. I recommend it. It's about a mile hike in from the road, very easy to get to. So can you imagine life starting up in something like this? Well, I'm going to claim that this is the kind of condition that prevailed on the early Earth, and that the conditions, the physical, chemical conditions of such an environment are what would have given rise to the polymerization processes that finally gave rise to living cells. Charles Darwin was thinking about the same thing long ago. Here's a little note he put in a letter to his friend J.D. Hooker, 1871. But if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. So wasn't that a prescient thought? This guy was smart. He was looking ahead to what we're doing right now. Thinking about warm little ponds, I think about hot little puddles, thinking that something like proteins, a polymer could be formed, and my hat's off to Uncle Charlie for thinking that and writing it down. Here's what I think Charles Darwin's warm little pond might look like now that we know more about it. It would have been in a volcanic setting, such as I showed you at Bumpus Hell. Here's one in a very famous painting in the basement of the Smithsonian, what the Earth was like several billion years ago. So here's a warm little pond, a la what we know now. And I decided to go to Kamchatka. The Bumpus Hell is right in the middle of a lot of plant life and a lot of people tramping by. I wanted to go to a sterile environment, still an analog environment, and such an environment is in Kamchatka, Russia. I've been there twice. The Kamchatka geothermal regions I consider to be what we call primitive Earth analog environments. High altitude, high latitude, and volcanism produce sterile sites for experimental tests. Can self-assembly occur? This is what Kamchatka looks like. If you go 2,000 miles east, you're in Anchorage, Alaska, which is where we fly out of to get here. We land at Petropavlovsk, little upper arrow there. We hop in a Russian troop carrier, and 5 hours later, we are delivered to Mount Mitnovsky, which is the little bottom arrow there. Here's one of our trips. Some of the people in the audience know some of these people. Jan and Jake are well-known astrobiologists now. They were postdoctoral people at the Carnegie Institution of Washington. Jamie Elsila and Jamie Elsila and Megan Spencer were graduate students right here at Stanford University in Dick Zayer's lab. Jamie is now working in astrobiological problems at Goddard. Then we have Vladimir in the white cap, white coat. He was our local organizer. And clear to the right is Igor, who sort of was our guide for this trip. And clear to the left is my buddy and neighbor, Tony Hoffman, who is the photographer on this little field expedition. Here we are climbing up. It's about a 2,000-foot climb from this little hut we stayed in to the crater. Here's Vladimir looking down into the crater of Mount Mednovsky. And when I think about the primitive Earth, this is my vision of it. Sterile, nothing's there. Very dilute organic solution of stuff, that might be there from infall, from synthesis. Hot water, steaming volcanism all over the place, and little ponds that we can use as experiments. Here's a fumarole, and I've reached down into the fumarole just to test whether any organic material was there, anything alive. And I pulled out a flask full of mud. We took this back to my lab here at Santa Cruz and analyzed it, and really, there was nothing there. We had no indication of any organic material and no indication for sure of anything alive. So this is a good place to do the experiment I'm going to show you. So I revised Darwin's conjecture and said to myself, but if, and oh what a big if, we could conceive in some hot little puddle with all sorts of prebiotic amino acids and nucleobases, lipids from membranes, phosphoric salts, light, heat, electricity present. So that is really the experiment that I'm going to tell you about. Here's the recipe. This is what we're going to put into a hot little puddle. We're going to have 4 amino acids, 4 nucleobases, 1 gram each, phosphate, glycerol, myristic acid, that's this thing here. It's a amphiphilic soap-like molecule. That's our membrane-forming material. Then we can just pull out samples every hour for several days, and we're going to ask whether self-assembly can occur and whether any interesting synthesis can happen. And here's what we found out. Let me just show you what it looks like. This is Darwin's hot little puddle. That's a boiling area right in the center. That's about 93 degrees at this altitude. And this is clay down, well, yeah, here's the clay is down there. So a lot of volcanism produces clay. And up there, we have fluctuating wet-dry cycles. And keep that in mind. I think that's really important, because at the edges of pools like this, it's sometimes going to get wet, sometimes going to get dry, wet, dry on an hourly basis, daily basis. So this is a place where interesting chemistry can occur. And what we're going to ask first, though, is can self-assembly of membranes occur? So here I am pouring a primordial soup, prebiotic soup, into Darwin's hot little puddle. Notice it's white. The reason it's white, same reason this is white. It's got membranes in it. It's got the lipid, the myristic acid. And what I want to show you now is the same thing about an hour later. And with apologies to Michelangelo and the Sistine Chapel, I think that this is where life began. So that white stuff that you see, the white scum, is self-assembled membranes. So I'm satisfied that I can make at least compartments in a thing way outside the laboratory. This is very comforting to me anyway, and my colleagues think I'm a little bit crazy for doing this sort of thing, but that's okay. I'm at a point I don't mind being a little bit crazy anymore. So here we are now with this membranes, and what we want to do now, though, is to see if the membranes can do any good in terms of polymerization. So that brings me to the last part of my talk. So this shows you close up what we know membranes are produced by at the molecular level. Down at the bottom, you see a phospholipid and a cholesterol. And this is what is in the membranes of every cell of your body. 100 trillion cells, every one of them has a membrane around it made out of those 2 components. This is what the prebiotic membranes are made out of. There's that fatty acid or soap that I've been telling you about. So this is what a lipid bilayer looks like close up. This is a model from Andrew Poharil right here at NASA Ames, who does this kind of modeling. And you can see that there is a oily center. This is a cross-section through the membrane. There's an oily center composed of hydrocarbon chains, hydrophilic groups on either side in contact with water. So that's what a membrane looks like, the lipid component. So keep that in mind because you're going to see that again as we go on. Last point to make before I get to the molecular biology is that a property of vesicles is that when they fuse, they turn into a multilamellar matrix, what we call a lipid matrix array. And anything that is outside becomes trapped between those layers. You can see them trapped, the little sort of pink lines there. When you rehydrate, you get the stuff back, and now it's inside the vesicles. Some of the vesicles have stuff inside. To prove that to you, here's a image of what we call freeze fracture electron microscopy. Here's some vesicles on the left. You can see they're about the size of bacteria, 2 microns. When you dry them, they turn into that dry multilamellar lipid phase, and that is what we believe is able to trap anything that's outside gets trapped between those lipid lamellae that you see on the right. To prove that to you, we have put some DNA in with some vesicles, put it through a single dry down rehydration. All those bright fluorescent things is stained DNA inside the vesicles. It was all outside to begin with. So it just demonstrates how powerful it is to put it through a single wet dry cycle. You get these protocells. Same thing I just did for you here. Okay, now a little bit of biochemistry. I'm going to try to keep it as simple as I can for those of you who haven't had biochemistry. This is adenosine monophosphate. It's one of the 4 monomers of DNA and also one of the 4 monomers of RNA. That's what it looks like. The blue and black thing upper left is adenine. The black and red thing in the middle is ribose. And the little red and yellow thing is phosphate, adenosine monophosphate. That is one of the subunit monomers of all nucleic acids. So what we're going to try to do is to get that to polymerize under these conditions I've been telling you about. Here's a cartoon of what we think is happening. There's the lipid bilayers I showed you, and look at that little thin line. Those are all of the AMP molecules that have been flattened out and organized between the lipid lamellae. And they're not just stuck there, by the way. They're able to diffuse around because this is a liquid crystal. This is not solid. These things are able to diffuse around in 2 dimensions. So here's what I claim. I claim that every time 2 of those come together, there is a chance for a water molecule to leave and produce what is called an ester bond. That's what holds nucleic acid together. Ester bonds, very simple. Pull out a water molecule, you make a chemical bond, and that, if you make it long enough, you got a DNA molecule or an RNA molecule. So the question is whether this organization can help that process along. Just another view of it. There is up on top, I just show you a solid glass where you just dry the AMP down without any lipid present. It forms a solid glassy phase. Can't react because everything's stuck in place. There's no diffusion. But if we do the same thing and organize it in a lipid matrix, what can happen is that water can leave and the stuff can polymerize. So that's the idea. Now, if there are any organic chemists in the audience, will be of interest to you, almost to nobody else. This is that mechanism that I told you for ester bond formation. Basically, we're reversing hydrolysis. Now, in hydrolysis of RNA, water is added to ester bonds, and you break it down to the mononucleotides. And all we're doing is setting up conditions where water can leave the mononucleotides and produce RNA. Very simple. This is called Fischer Esterification, discovered 100 years ago by a very famous German organic chemist. There's a bunch of little resonance structures. The main thing is down at the bottom, you get a ester bond, water leaves, and you can make a ester linkage. Does it work? The answer is yes, it does. In 2008, we published this paper, and in it we showed that if you dry down vesicles with monomers and cycle it up to 7 cycles, wet dry cycles, at an elevated temperature, 85 degrees C, under these conditions, you can make RNA molecules. Notice that upper left, you can see cycles, 1, 3, 5, 6, 7. Each cycle produces more and more of this stuff. And by the way, that black stuff is radioactive label, and we've attached that to the end of these RNA molecules with a little enzymatic step that you see off to the right. RNA in yellow at the top, those enzymes add a radioactive phosphate. The radioactive phosphate shows up in these gels that we can use to separate the product. And notice also what we call a ladder. It's 10, 20, 30, 40, 50, 100. Those are known lengths of RNA. And if you look up at the 100 and bring your eye across, some of those molecules we made are way up in the 100-mer range. This is really long, plenty long enough to make a ribozyme, for instance, out of it. So everything else you see there is individual experiments, very reproducible. We've just varied the conditions, and it's not very interesting except to say that it's a robust reaction and works. Okay, here's the last stuff I want to tell you about, just a few more slides. How did replication begin on the early Earth? No one knows. We just don't have a way to think about this yet. But I think we have a lead into this. Maybe replication can occur under the same conditions that I've talked about. So we tried this out, and a paper was published early this year, paper by Felix Olasagasti. He's from the Basque region of France. Kim and Pourmand. I'm the only American citizen in that particular team, which is very common in university science. And look what we're claiming. Non-enzymatic transfer of sequence information under plausible prebiotic conditions. That is a big, scary claim. And until somebody repeats it, I don't want you to take this seriously. We're the only people that have done this so far. We're hoping that other labs will pay attention and repeat it. If you're interested, it came out in Biochimie 2010. It's very late in the year. Here's the idea. If you have a solution of the 4 bases, A, G, T, and C, those little colored squares, in the presence of a template, and if all you do is dry it down, you get a glassy solid. Not much can happen because it just it dries out, the stuff cannot diffuse around, it cannot line up by what we call Watson-Crick base pairing. But if you dry it in the presence of lipid, the stuff can move around. It has enough time to find the complementary base pairing between the yellow and blue, which is A and T, and the red and green, which is the G and the C. As you see them all lined up there. And then, because these conditions allow ester bond formation to occur, you zip up all of those ester bonds and you end up with a double-stranded DNA that has transferred the sequence from the template to the product strand. So here's the formula for doing this, so a little recipe. Pretty simple. We're just simulating a fluctuating environment, such as I've shown you. We mix the DNA template about a 55-mer. That means it's got 55 nucleotides. We know the sequence. It's got 4 nucleotides, A, T, G, and C. We have phospholipid vesicles. That's the stuff that is going to form that multilamellar matrix. Going to dry it down under carbon dioxide. Oxygen tends to inhibit this. Going to give it about 2 hours at 85 degrees. We're then going to add water, dry, heat again, repeat 5 times. Then we're going to see whether there's any double-stranded DNA. If we're right, we should see double-stranded DNA there. Furthermore, if we see it, we can be able to ligate the products together, sequence the products, and see whether any information is transferred from the template to the product strand. And that's what we did. That last bit, by the way, is very high-tech sort of molecular biology, and I won't have time to go into it. Then we got to compare, and use bioinformatics techniques to compare the base sequence to the expected sequence. Okay, DNA is synthesized. See that thing there says complete system? This is a fluorescence assay for double-stranded DNA. When you have the complete system, you get double-stranded DNA out of it, as indicated by this fluorescent dye. We then took the material and put it onto a gel. We did electrophoresis and see that little, clear to the right, see 50 base pairs. That is in the range of our template. And you can see that we have a double-stranded DNA showing up in the gel in that 50 base pair region. So we're pretty sure now that we've not only made double-stranded, but that we can separate it. So we've isolated that double-stranded material, ligated it, did a sequence analysis, and finally compared alignment scores. If all we had made is random fragments of DNA, it would have shown up on the left side of these alignment scores. In other words, virtually no alignment at all. As the alignment score increases, that means that we have sequence information transferred. So all of that product that you see there is our product sequence. And those are big alignment scores. They're up at the point where we have only about a 10% error in transfer of information between strands. So that is the main thing you should take away from this. We know that adding lipid gives sufficient complexity to allow RNA synthesis. They become ordered within that array that I showed you. If a DNA template is present, we can get sequence information transferred. The next question is how to get this system to evolve. Nobody knows how to do that yet. That's where we are. We've had a few steps, though. Jack Szostak, my colleague back at Harvard, has taken an interest in this. He's gotten a partial DNA to occur inside a fatty acid vesicle under conditions where an activated monomer can get across the membrane, add itself to that partial DNA to complete the double helix, and then those large vesicles can divide into smaller vesicles. So that's about where we are now in the search for how life began and how to simulate it in the laboratory. Thanks for your attention.